Today we are talking about vitamin D. When I say vitamin D, what's the first thing that pops into your mind? Bones, sunlight, maybe that calcium supplement your grandmother takes? Excellent. But what if I told you that this vitamin is actually a sophisticated pro-hormone, a master conductor orchestrating a symphony of physiological processes far beyond just bone health? We'll uncover how this seemingly simple compound transforms into a potent steroid hormone, wielding influence over immunity, cell growth, and even your mood. This isn't just about passing your exams. It's about understanding a molecule critical to the health of every single patient you'll encounter. From a gaon ka bacha with rickets to an elderly dadi with osteoporosis. So grab your virtual chai, settle in, and let's decide the enigma of vitamin D. First and foremost, why does this matter, especially in India? So, here I like to discuss the paradox of plenty. So, India, the land of abundant sunshine, ironically has one of the highest prevalences of vitamin D deficiency globally. This isn't just an academic curiosity, it's a significant public health challenge that affects millions, often silently. Despite our tropical latitude, factors like lifestyle, cultural practices, and even air pollution play a crucial role in the widespread deficiency. There's a journey that vitamin D undertakes. Okay, it begins with a familiar steroid, a derivative of cholesterol, setting the stage for a fascinating transformation. It all starts in the skin, where there is molecular sunbathing in a way. It starts with the precursor molecule, where the story kicks off right beneath your skin, specifically in the epidermal keratinocytes. Here we find 7-DHC, which is short for 7-dehydrocholesterol. This isn't some exotic foreign molecule. It's a perfectly normal intermediate in your body's cholesterol biosynthesis pathway. It is strategically positioned in the cell membrane for its solar density. Think of it as a raw roti dough, perfectly shaped and waiting for the tawa, which will be the sun, to transform it into something nourishing. The sunlight acts as a catalyst and it initiates a photochemical reaction. So, when your skin is exposed to ultraviolet B radiation, or in short UVB radiation, particularly wavelengths between 290 and 315 nanometers, this 7-DHC absorbs this energy. This is a critical point. This isn't an enzyme catalyzed reaction. This is pure photochemistry. So, unlike so many of the reactions which we see in biochemistry, this is special in the way this is a photochemical reaction and not an enzyme catalyzed one. The UVB energy causes a specific carbon-carbon bond in the B ring of 7-DHC to break. The UVB energy causes a specific carbon-carbon bond, specifically C9 and C10, in the B ring of 7-DHC to break, essentially opening up the steroid ring structure. It's like the sun's rays are a magic key, precisely unlocking a biochemical transformation in a non-enzymatic fashion. Now, as a side note, there are certain factors affecting skin synthesis. Some of them I'm going to list out here. The first one is the time of day or year. Okay, so midday sun, typically 10 a.m. to 3 p.m. in summer months, is most efficient due to the high intensity and direct angle of UVB rays. In many Indian countries, in many Indian cities, particularly during winter or periods of high pollution, we call that smog, Atmospheric scattering and absorption can significantly reduce the effective UVB reaching the skin. Next is latitude. So closer to the equator, more direct UVB exposure is possible year round. India is generally well placed geographically, but northern regions during winter months might experience insufficient UVB intensity for effective synthesis. Next is melanin content. Darker skin tones, which are common across India, have higher concentration of melanin. Melanin acts as a natural broad spectrum sunscreen, absorbing UVB radiation and competing with 7-DHC for photons. This means individuals with darker skin require significantly longer sun exposure, up to 5 to 10 times more, for equivalent vitamin D synthesis compared to fairer individuals. This is critical factor contributing to vitamin D deficiency in our population. Then this clothing and lifestyle. Traditional Indian attire like sari, salwar kameez, or burqa in some communities often covers large parts of the body. This limits skin exposure to the sun. Furthermore, increasing urbanized and indoor lifestyles common among students and professionals drastically reduce opportunities for adequate sun exposure. Then there's age. As we age, the concentration of 7-DHC in the skin and the skin's overall capacity to synthesize vitamin D3 decreases significantly, making the elderly particularly vulnerable. Okay, so now that we have discussed a couple of factors influencing the vitamin D production, next let's move on to the product formation. Okay, this initial photolysis which has just occurred generates pre-vitamin D3, which is also called pre-cholecalciferol. This molecule is kinetically unstable and transient, a bit like a gulab jamun, fresh out of the hot oil, delicious but unstable. Next is isomerization, or the cooling down. So this pre-vitamin D3 is thermally unstable over a period of hours, typically 24 to 48 hours. At normal body temperature, which is 37 degrees, it spontaneously undergoes a heat-dependent isomerization. This involves a rearrangement of its double bonds and the formation of a more stable, biologically active form, which is vitamin D3 or cholecalciferol. This is a crucial non-enzymatic temperature-dependent step, ensuring the stable product is formed without further UV exposure. So this is another unique point in the vitamin D metabolism where you're seeing the step is non-enzymatic. So think of 7-DHC as a raw papad. UVB light is the fire that roasts it, transforming it into pre-vitamin D3, which is a slightly burned, still hot papad. Then, with a little cooling and time, it settles into a stable, crispy papad, which is vitamin D3, ready for consumption by your body. So this was the step 1. The step 2 is the dietary intake. So not all vitamin D comes from the sun and the skin. We also obtain it from our diet. Vitamin D3, which is cholecalciferol, this form is primarily found in animal-based sources. Rich sources include fatty fish like salmon, mackerel, and sardines. Cod liver oil and, to a lesser extent, egg yolks and liver are now fortified with vitamin D3, which is a significant public health strategy. Vitamin D2 or ergocalciferol. This form is derived from ergosterol in plants and fungi. Example, UV exposed mushrooms. It is also used in some fortified foods and supplements. So here's an interesting point for you. Both D2 and D3 are biologically active precursors. However, vitamin D3, which is cholecalciferol, is generally considered more potent and stable in humans, with a longer half-life and greater affinity for the DBP. For our largely vegetarian population in India, dietary sources of D3 are particularly challenging, making fortified food, supplementation, and conscious sun exposure even more critical. Now, there's another step, which is the transport to the liver, which I also like to call the auto rickshaw ride. So, whether synthesized in the skin or ingested, vitamin D3 and D2 is lipophilic, meaning it's fat soluble and doesn't mix well with the watery environment of your blood plasma. It can't just float freely in the bloodstream like pani puri in its pani. It needs a ride. It's a transporter bound to a specific high affinity carrier protein, which is called the DBP, the vitamin D binding protein. DBP is a liver synthesized alpha 2 globulin. It ensures its solubility, protects it from premature degradation, and delivers it efficiently to target organs, primarily the liver. Think of DBP as the reliable auto rickshaw driver, ensuring our vitamin D reaches the first important metabolic destination safely and efficiently. DBP circulates in great excess, meaning typically less than 5% of its binding sites are occupied. The next step is the first hydroxylation. This is the liver's moher, which is the stamp of approval. So this occurs basically in the liver. The first crucial metabolic step, essential for activating the prohormone, occurs predominantly in the liver, specifically within the hepatocytes. The enzymes are located in both the mitochondria and microsomes. The enzyme which changes the vitamin D3 is the 25 hydroxylase enzyme. It leads to hydroxylation of the vitamin D3, and it is a CYP2R1 cytochrome P450 enzyme. Other P450 enzymes such as CYP27A1 can also contribute to 25 hydroxylation, particularly in extrahepatic tissues, but CYP2R1 is considered the primary physiological enzyme in the liver. So this CYP2R1 is a monooxygenase. It uses NADPH, if you can recall from the pentose phosphate pathway, as an electron donor and molecular oxygen to add a hydroxyl group to the carbon at position 25 of the vitamin D molecule. This reaction is largely unregulated by the body's calcium and phosphate status, meaning the liver will hydroxylate most of the vitamin D it receives, creating a circulating pool. So, cytochrome P450 enzymes are a superfamily of heme-containing monooxygenases, vital for metabolizing a vast array of compounds, including drugs, steroids, and xenobiotics. They catalyze the insertion of one atom of oxygen into a substrate, while the other oxygen atom is reduced to water. This process requires reducing equivalents, typically from NADPH, and involves a complex electron transport chain. So yes, this is an energy demanding process, but absolutely crucial for the initial activation of the prohormone. The final product is 25-hydroxy vitamin D, or 25-OHD, also known as calcidiol or calcifidiol. This is the main circulating form of vitamin D in the body, and due to its relatively long half-life, which is around 2 to 3 weeks, it is the form we routinely measure in blood tests to assess a patient's overall vitamin D status. It has some minimal biological activity as well, but it is largely considered a prohormone, which means it's awaiting its final tightly regulated activation. So think of 25-OHD as a partially charged mobile phone. It's got some power, but it's not fully armed or ready for its most important functions. This is the mohair stamp from the liver, making it identifiable and circulating. So the next step is the second hydroxylation, which occurs in the kidneys. So this is arguably the rate limiting and most tightly regulated step in vitamin D activation. It occurs in the kidneys, especially within the mitochondria of the proximal tubule cells. While the kidney is the major endocrine producer, Some extrarenal sites, example macrophages, placenta, prostate, breast, colon, skin, can also express this enzyme, allowing for local calcitriol production for specific paracrine or autocrine functions, example local immune modulation. But the kidney controls systemic calcitriol levels. So the enzyme here is which is a star enzyme, is 1 alpha hydroxylase, also known as CYP27B1, another mitochondrial cytochrome P450 enzyme. This enzyme is a crucial gatekeeper controlling the final active hormonal form of vitamin D. Its activity is exquisitely sensitive to the body's need for calcium and phosphate. So the mechanism is basically that CYP27B1 catalyzes the addition of a second hydroxyl group, this time at the carbon at position 1 of the 25-OHD molecule. Like CYP2R1, it's a monooxygenase that consumes NADPH and oxygen. The precise and intricate control over the activity and expression of this enzyme is what truly elevates calcitriol to the status of a hormone. So the final product is 125 dihydroxy vitamin D, or 125 OH2D, also famously known as calcitriol. This is the biologically active hormonal form of vitamin D, responsible for mediating most of its well-known physiological effects. So calcitriol is the fully charged mobile phone, connected to the network, ready to make calls, send messages, and navigate. It's the chabi, the key that unlocks the body's calcium and phosphate machinery, initiating a cascade of effects. The next step is catabolism and inactivation, which helps in maintaining a balance in a way. To prevent toxicity and maintain precise homeostasis, which is your body's balance, active calcitriol is rapidly inactivated by another crucial cytochrome P450 enzyme, which is 24 hydroxylase, or CYP24A1. This enzyme is widely expressed particularly in the kidneys, but also in many other VDR expressing tissues, serving as a critical negative feedback regulator. So CYP24A1 initiates the catabolic pathway by adding a hydroxyl group at position 24. This leads to the formation of 24-25 dihydroxy vitamin D from 25 hydroxy vitamin D, or 124-25 trihydroxy vitamin D from calcitriol. These products are then further metabolized through a series of oxidative steps to inactive water-soluble forms such as calcitroic acid, which are ultimately excreted primarily in the bile. This is a crucial negative feedback mechanism. High calcitriol levels directly induce the expression of CYP24A1, thus accelerating its own breakdown and reducing its active concentration. It's like the body having a built-in off switch or a traffic controller to prevent excessive accumulation and potential toxicity. Coming to the next step, the mechanism of action, or the molecular switchboard. So active calcitriol, which is 125 OH2D, acts as a classic steroid hormone. Unlike peptide hormones that bind to cell surface receptors, calcitriol, being lipophilic, which is fat soluble, easily diffuses across the cell membrane to enter target cells. Receptor binding occurs inside the target cells, where there's a high affinity to the vitamin D receptor, or the VDR, which is a specific type of nuclear receptor. VDRs are found in the cytoplasm and nucleus of cells in numerous tissues throughout the body, including intestines, bones, kidneys, parathyroid glands, immune cells, and even various cancer cells. Upon binding calcitriol, the VDR undergoes a conformational change. This change allows it to form a heterodimer which is a complex of two different protein units. It forms this heterodimer with something called RXR, retinoid X receptor, which is another nuclear receptor which binds to the retinoic acid, which is a derivative of vitamin A. This VDR or RXR heterodimer is the biologically active transcription factor. It translocates to the nucleus if it's not already there and binds to specific DNA sequences called vitamin D response elements or VDREs. These VDREs are located in the promoter regions of target genes. They are like specific address codes or command prompts on the DNA indicating which genes are regulated by vitamin D. This binding recruits coactivator proteins and either initiates, upregulates, or represses the transcription of specific genes. This leads to changes in messenger RNA production and subsequently altered protein synthesis and cellular function. So, examples of gene regulation include calcitriol upregulating genes for proteins involved in calcium absorption in the gut. Example, calbindin, TRPV6 calcium channel, or calcium ATPase. Calcium reabsorption in the kidney and proteins involved in bone remodeling like osteocalcin or rankel. It also influences genes involved in immune function, cell cycle control, and insulin secretion. So calcitriol is like the CEO's order that reaches the specific department, which is VDR or RXR, which then activates or deactivates certain production lines, which are genes, to achieve a desired outcome, which is protein synthesis. This precise gene regulation underscores its role as a powerful hormone.